Guys, greetings. Happy New Year. I am starting this video with a very brief introduction as I committed to you that this is going to be educational videos with basic fundamentals, approach to understand the concept, get deeper insights, as well as get some ideas that how you are going to have a research background in this area. This topic will also help you to gain knowledge, build confidence, and gain insights deeper into that concept. Today I am going to talk about a topic in which I am involved for the last 10 years as a topic of research, and this term is capacitation. I hope everyone knows what is capacitation, and this is not a new word. This word was started in the year 1952 by Austin. It was discovered in the year 1951 by Chang and Austin independently. Chang described this concept in rabbits and Austin described the concept in rat. It was outstanding phenomenon in that time because it was scientific aspect. No one is having that kind of instruments or equipment set up to see it in vitro, in vivo. But it was the scientific temperament of those scientists and commitment which discovered this concept. A very famous experiment were conducted independently by these two scientists, and later on it emerged as one of the most powerful areas of reproduction. So before going into the details of this topic, let us go through a preview of this word capacitation. What actually it means, why it is so important, and why it is deeply associated with the sperm biologist. Now, this is a phenomenon which is very much important and significant for the IVF, and it is always a challenging process for the sperm to get capacitated. So literally speaking, capacitation is a process. It's a complex group of functions. It's a complex group of events. It involves extracellular signaling molecules, intracellular environment, membrane association, downstream molecules which are associated deeply and intensively in regulating this process. Now coming to this word, it is the ability of sperm cells to undergo physiological and biochemical changes so as to acquire the capacity to fertilize. It's a very wide definition. This definition says multiple words. Ability of the sperm cells. I can say this is also a process in which the spermatozoa is undergoing, or the sperm cells are undergoing, a number of physiological changes. We will discuss that. Number of biochemical changes. Again, we have to discuss in details. So as to acquire the capacity of fertilize. It means now these words are putting big question marks. What is acquire? What do you mean by physiological? What is the meaning of biochemical? Capacity. What do you mean by the capacity or ability? It means these words are making now this word capacitation a very interesting topic. It means I want to say, now if you go to the gonads, that is the testes. These are the gonads. Gonads means the structures where actually the gametes are produced. They undergo hormone and genetic dependent mechanisms to produce the gametes. And the male functional gametes are known as the sperm cells, which are produced in the male gonads known as the testes. In the testes, the process is known as spermatogenesis. It's a complex process. It's a thoroughly regulated, precise mechanism which occurs in the germinal epithelial cells. Gradually, the process becomes more complex when division, maturation and growth phases occur, Eventually, you are getting the cells which are haploid, which are having half set of chromosomes, and they are known as the sperm cells. So spermatogenesis is the process in which the sperm cells are formed inside the testes. It's not important that they are formed inside the testes and they are having the ability to fertilize. Rather, they will undergo a number of, or you can say a series of mechanisms in epididymis, another organ where the spermatozoa undergoes storage, maturation, a phase of quiescency. Metabolic activities are almost down, and the sperm undergoes a number of processes by which it makes it competent, makes it capable to undergo the process further development and undergo the process of fertilization in the female body. So epididymis is the structure where the sperm spend a lot of time. It varies on the species. It may be from some days to few months, and thereby it acquires the capacity to fertilize. So in the epididymis, the spermatozoa undergoes modeling. And once modeling is over, then it gets maturational changes. And what are those? This will come in my next video when we will talk about the epididymis, functional role of epididymis in regulation of the sperm function. And once modeling is over in the epididymis, then the spermatozoa undergoes this process that is known as ejaculation. In the coming videos, we will discuss in extensive all these processes in detail so that we can understand how these events occur. And once ejaculation is over, the spermatozoa comes into the female body. So now very important question. Are the sperm from the testes are fertilizable? Yes or no? Number 2, are the sperms which are coming to the epididymis are fertilizable? Another question, yes or no? And whether the sperm which are directly ejaculated into the female body or inseminated into the female body are said to be fertilizable? These are the question mark. With the advance of the science, we can say that we can model them, we can simulate them, we can provide conditions to them by which the sperm acquires the fertility. But in general, naturally, these sperm are not fertilizable. And that was the point in 1951 when the Chang and Austin was throwing the insights into this word capacitation. It means in a nutshell I can say, or the sperm which are coming from the male body, from the testes or in the ejaculation are only fertilizable if they undergo number of changes. These are physiological, biological, biochemical, number of surface changes, number of intracellular changes. Thereafter, they are able to bring out the process of fertilization, and that's what known as capacitation. So, before going into further details, let me tell you two very good stories of the science. And these famous 2 experiments change the concept by which we are understanding or we are seeing the process of capacitation. And it was the very famous experiment which was conducted by Chang in rabbits. What he has observed, how this word came into picture. Now Chang selected the rabbits, selected the spermatozoa to be inseminated, and then he observed that once the ejaculated sperm, after dilution, when they are inseminated into the female body, they are not able to bring the fertilization immediately. Rather, the sperm which are exposed to the female uterus or female microenvironment in the genital tract, they are becoming more fertilizable. Around 2 to 4 hours, 4 to 6 hours, in differential condition, in optimized conditions inside the female body, they acquire the fertilization. That's why the term came into picture. The term was, he was not the person to coin this word capacitation, but this was the first systematic study which was also published in Nature in 1951. Then another independent scientist, that is Austin, he carried out the study in the rats. The similar modeling studies, but he also marked that the female rats, they are going to be fertilizable, means the oocyte is going to be fertilizable if the sperm cells are exposed to the female body around 2 to 4 hours. And these conditions where the spermatozoa stay in the uterus or going towards the oviduct, they are undergoing number of modeling reactions. And these modeling reactions are known as capacitation, and that make the sperm fertilizable. So in the year 1952, Austin was the man to coin the word capacitation. From 1952 to 2025, thousands of research has been done. And very proudly I am saying from 2014-15, I am also deeply involved in understanding this event of capacitation. Let me tell you very few interesting things about capacitation which are not there in the generalized book. We think that when we are having an ejaculated semen, we expect that all the sperm will undergo the process of capacitation and they will acquire the capacity to fertilize. But this is wrong. Not all the sperm in a population are going to be capacitated. It is a number of sperm. You can say it is a population of sperm, or you can say it is a subpopulation of sperm which only respond to the female environment, the female constituents, and they undergo the process of this capacitation. So this is a big question mark for the sperm biologists to understand how these events occur. Why not all the spermatozoa are at a time capacitated? This means this process is really complex. This process is selective, dynamic, and it occurs only to those sperm which are really undergoing the process of remodeling inside the female genital tract, and especially that is in uterus. But the real signaling mechanisms occur in the oviduct. When the sperm reaches there and the oocyte is present, the oocyte is sending the signal, sperm is acquiring that signal, and these mechanisms I will discuss in vivid in my coming videos. But for the time being, we should understand the sperm which are coming to the uterus, staying there for 2 to 4 hours, 6 hours, 8 hours, depending on the species, they acquire the capacity, and that capacity we are explaining as capacitation. Another very famous scientist, Hammond, he was the person to carry out independent studies, and also these three scientists, Chang, Austin and Hammond, were conducted the pioneer studies during 1950s and 60s. So what we are studying today is the outcome of those classical experiments, and now with the advance of the science, imaging technique, molecular biology, single cell imaging, we are now clearly understanding how actually the capacitation is occurring. So the second interesting point is that if the spermatozoa are same in the semen and if they are ejaculated in a mass, why they are not undergoing the capacitation as a whole? This is a big question. And the answer, somewhat, I can say that semen is a homogeneous population of cell, is a heterogeneous population of cell. So studies say that semen is a heterogeneous population of cells. That means even we are saying that a semen ejaculate contains similar number of cells or similar number of sperm cells. No, they are not. They are morphologically distinct. They are physiologically distinct. Maybe they are appearing anatomically similar, but their cellular machinery which are present in them, they are said to be different. And that's why their response to the microenvironment of the female, to the hormones, to the agonist, to the molecules, to the ligands are said to be different. And therefore, the mechanism which occurs inside them are also said to be different. The signaling cascade which are activated inside them are also said to be different. And therefore, the result ultimately which comes that a group of cells, a small group of cells, or a small population of the cell, or a small subpopulation of the sperm cells getting capacitated. So that makes this process very interesting. When we are saying that in the female genital tract, millions of the spermatozoa are coming, then why only few are said to be capacitated? This is another very interesting question. Scientists are trying to understand this process. They are trying to solve this process so that strategies can be developed so that conceptual failures should not be occur or fertilization failures should not be occur. So giving this overview to students or those people who are seeing this should understand that this is not a small world. This is a world itself. And this world is the primary basis of a sperm to carry out the process of fertilization. So this we should understand. And when I am saying that ejaculated spermatozoa are coming, staying, spending some time, and that is the quality time for the spermatozoa, I can say. And in that time, there is a crosstalk occurs. The sperm also talks with the uterus, uterine fluid, uterine cells, uterine secretions. The sperm also talks with the oviductal epithelium, oviductal cells, oviductal secretions. They also talk with the oocyte which is ovulated. And altogether this makes this phenomenon very complex. It's not a unitary pathway that we can understand at one point, Rather, it is involving multiple pathways, and that makes it very complex. Even today, reproductive biologists still try to understand what exactly happens, what switching on or off mechanisms occur so that the sperm are getting the acquisition capacity to fertilize. So this makes this introduction over. Now I will draw a flowchart to show a beautiful journey of the spermatozoa from the male body to the female body. But along with that, I need to say that this is not an easy journey. This is a complicated journey. Number of complex obstacles are given to the sperm, but the sperm are so competent, so dynamic, they cover all the obstacles, try to reach to that point where they can meet the oocyte. Nevertheless, they are going to have those interactions with the oocytes, and ultimately that process will result. That's what known as fertilization. So once again, I will say the definition as it is the spermatocells. They undergo number of physiological and biological changes, biochemical changes rather, to make it capable for the process of fertilization. So we will discuss in detail, but see this flowchart which will say what actually capacitation is, how it occurs, why premature is not required, where a site-specific capacitation is required, what are the different events which occur in a phase-wise manner during the time of capacitation, who is playing the important role, is it the male secretions from the seminal vesicles, the prostate, or is it the secretion of uterus or the oviduct or else other thing. That means overall this is a very interesting topic. Let us draw another flowchart to see how the journey is so complex. And guys, one more thing. Sperm is the only cell of the animal kingdom which passes its life cycle in two different individuals, male body and female body. A sperm life cycle is going to complete only when it reaches the female body, where it will undergo this process known as fertilization. That means we should not forget how the sperm is dynamically evolved. Different pH, different osmotic gradient, different temperature, all these parameters make the sperm journey very complex. But even of that, the spermatozoa wants to meet his soulmate, that is the oocyte. And this process is regulated, dynamic, precise, controlled, and very complexly organized. So let us understand that. So it is the testis shows the spermatogenesis. Once the spermatogenesis is over, the sperm cells are produced. These sperm cells are said to be non-motile. They are said to be non-fertile naturally. I want to emphasize this, naturally. In artificial conditions, we can make them fertile, but naturally they are not fertile. They require remodeling. They require maturation. And all these processes occur in one structure that is known as epididymis. This is the structure which is present along the testis. That is epididymis. In the epididymis, I am continuing from here. In the epididymis, after this, the sperm cells come in contact with the seminal plasma. The seminal plasma are secreted from three different areas where the sperm is mixed. The number one is the epididymis, 10% of the secretions. The number two are the seminal vesicles. Around 60% of the secretions are mixed to the sperm. The third one is the prostate gland. It will form 30% of the total secretions. Once it interacts with the seminal plasma, then the sperm undergoes ejaculation. We'll discuss in details in our coming videos, but for the time being, this flowchart I am drawing to focus, to draw your attention how complex these events are. Now ejaculation is over. The semen is coming either to the vagina or to the junction of the cervix, and from where its real journey starts. We'll take another class where I will discuss how the sperm survives, how the sperm is remodeled, how the sperm is restructured. But for the time being, from the cervix, it goes to the uterus. And in the uterus, it stays 2 to 3 hours, 4 hours, depending on the species. Undergo number of crosstalk mechanisms. So that is our next class. So in the uterus, the sperm undergoes, I can use the word remodeling, I can say signaling, I can say several set of modification mechanisms. And all these are physiological, biological, biophysical, structural and chemical mechanisms. The sperm becomes competent now to enter its final destination, that is oviduct, the fallopian tube. In the oviduct, sequentially it moves. Isthmus, ampulla. Ampulla is the site of fertilization. Isthmus, uterotuber junction. So I'll draw another diagram to show how the sperm swims in that. But at the oviduct, now the signaling started. And this signaling is a bidirectional crosstalk. This signal is from the oocyte cumulus cells along with a progesterone gradient. This is the signaling with the oviductal secretions, oviductal epithelial secretions, ciliary secretions, motions, or it is the interaction with the fluid which is present in the oviduct, or it is the swimming mechanism which is acquired by the spermatozoa from the uterus, and ultimately this signaling leads to this process, capacitation. We'll discuss vividly in my next video, that is capacitation part 2. This is today the capacitation 1. Just a brief introduction how the sperm is going to be capacitated. And once it undergoes capacitation, what actually happens? That swimming motility or that screwing motility with which the sperm is moving with a progressive manner, now it acquires a whip-like motion. And this whip-like motion is very fast flagellar motility. Or you can say the flagellar motion will make the sperm bend. The angle of rotation increases. Later egg-shaped jerky movements occur. That is known as the mechanical form of this word capacitation. That's known as hyperactivation. And this hyperactivation now leads to make the sperm capable to move near to the oocyte. And once it reaches to the oocyte, again a plethora, an array of signaling occurs. That we'll discuss in our coming classes. And ultimately, the sperm undergoes this process, what is being known as a chromosome reaction. That means the sac-like structure which is present on the anterior part of the sperm head is known as acrosome. It's a Golgi lysosomal derivative structure which makes the sperm to fuse with the oocyte. This is the process in which the acrosomal sac containing the enzymes are exocytosed. They are released on the surface of the oocyte. It tries to penetrate through the oocyte layers. The oocyte layers, once it will penetrate, then the pronucleus of the sperm comes in contact with the pronucleus of the female. And then that word which is known as, I can say this is that word fertilization in a very wide sense. Guys, you can well understand, I am telling you so many things, but I am writing only the key words. So it says that this process is not an unitary process. This is a sequence-like process. It is event-wide process. And this event-wide process occurs in a very regulated and controlled environment. It is very easy to say that I am conceived or the sperm are said to be fertilizable. But in the real sense, these are very difficult processes. Very complex processes. So that's why I selected this word capacitation to start with. And gradually I will try my level best to come to each and every point of the sperm journey. We'll try to understand how biological events, how physiological events occur that make the sperm to get ready for the most important and vital process of reproduction, that is fertilization. It's not a process, it is the generation of life, it is the creation of life. So it is a mutual exchange? No, it is a mutual fusion. Is it the mutual fusion or it is a programmed fusion? I can say this is programmed fusion. A very famous line I use while I teach in the class, that every spermatozoa may have the passport. Every spermatozoa may have the passport, but it doesn't mean that every spermatozoa is having the visa to enter to a country. So these are those sperms which are controlled, which are selected, and they are the only cells which are destined to undergo the fertilization. It's not a process of randomization. It's not at all. It's a process of selection. Oviduct plays a very important role. Now the new studies says that oviduct has the ability to select the best sperm. It is not Jo Jita Wahi Sikandar. It is the process that everybody is not having the capacity to fertilize. It is those sperms which are capable enough to undergo a mutual process of signaling along the female genital tract are only capable of the process of fertilization. So students, with this introduction is over. My thrust was to introduce this topic, capacitation. The take-home message is that capacitation is the process in which the spermatozoa acquire the capacity to fertilize. They undergo a number of physiological and biochemical changes on their surface, inside their cytoplasm, inside their organelles, in the flagella, in the flagellar assembly, in the mitochondria, so as to make them capable to reach to the site of fertilization. Capacitation is the biological word, but the mechanical form of the capacitation is known as the hyperactivation. A whip-like movement, the flagellar symmetry changes, the sperm becomes very fast in motility, a curve or an angular motion occurs at the region of the head, and gradually the spermatozoa moves, swings fast, asymmetric way and reaches the oocyte. So thank you so much for your patient hearing. I will wait for your comments, I will wait for your suggestions, I will wait for your questions. You can write me in my chat and you can ask me any questions from this class. But my next class is the capacitation 2, where we will move from Chang and Austin and we will directly go to the concept which are known, which will be beneficial to you. The passages, the pathways, the signaling which occur in a sequence manner in the sperm, so that we will ultimately get this word capacitation and activation. How the sperm undergoes this capacitation, how the oviduct has evolved with suitable molecules, what are the role of ion channels, how the electrical events occur in the sperm membrane, what are those electrical events, how people are knowing these electrical events, what are the signaling processes, what are the signaling molecules, what is the role of albumin, what is the role of cholesterol, what is the role of membrane fluidity, how the membrane fluidity is helping in the process of channel activation, what are the different channels, what is the role of Caspar, what is the role of intracellular ions like calcium, why flagellar assembly changes, why dining changes, what kinematic motions occur in the flagella, how the flagella is showing asymmetrical movement, why and how the sperm are getting a huge amount of energy, a huge amount of ATP is coming, how the sperm is showing hyperactivation, what is the role of Caspar, what is the role of SLO3, what is the role of HB1, what is the role of Na plus H plus exchangers, and all together how pH is regulated, whether it is intracellular pH, whether it is extracellular pH, but the sperm with a structure which is going to show a balance. So how these events occur? Cannot you think that this is very interesting? I am taking my knowledge, my experience from research, from books, from my career I have spent in this area, but till now I am not able to understand how the capacitation occurs. But till this process occurs, a multiple concepts are there. So wait for my next video. I am going to give this second part of this capacitation in few days from now. Thank you so much. Have a wonderful day. Have wonderful learning, and whatever the questions you are getting, write down in the chat box. Let's discuss. This is not a platform where I am having any restriction. I am not having any time restriction. I am not having any syllabus. It's a platform where I am talking, you are listening, and you are saying me, and I am listening. So this is a bi-directional crosstalk. So I want all of you to go through this lecture, read the literature, have any questions, ask me freely. Thank you so much. Have a wonderful day. Thank you.